Yeah, so choosing genetics and genomics, like a journey into a future shaping scientific career. So my journey began like the word genetics. So first, like I should really explain that thing. Like the word genetics, first, like I came to know about this word when I was 12-year-old. So for a 12-year-old child, this word seems to be a very novel word. So when I was watching a movie, and the male protagonist, I mean the female protagonist in that movie was a genetic engineer. So I was wondered, like in the entire screenplay goes something like this: an ancient DNA incorporated into a descendant, and he finds a cure for a contagious disease. And this was something which was thought provoking at the same time very much interesting. And then when I reached my 12th standard, this particular chapter, molecular basis of inheritance, this was one chapter which really like fueled up my brain with all these new terminologies like replication, transcription, translation, and I was a bit more fascinated with all these intricacies. So basically, it goes from a very like minute curiosity to choosing genetics and to discovering genomics and now like dreaming of immunogenetics. So why I chose genetics, so particularly this particular like initial spark of genetics was there embedded in my heart. And when I reached my 12th standard, obviously, as I told before, like this molecular basis of inheritance was one such chapter which I was really focused upon and very much interested in. And my biology teacher was extremely good in explaining things in very similar manner that even a small child could understand what is meant by central dogma principle. And then right after my 12th standard, everybody used to ask me the same question, like which subject you are going to take for your career. And everybody was pushing me to move for in the field of medicine and professional degree courses and all. But my mind was stuck up in something which was earlier being embedded in my heart, that is genetics. So in the year 2018, right after my passing out from higher secondary, I applied for BSc genetics, but unfortunately, God had other plans because due to an application error, I couldn't get into BSc genetics that year. So I went for medical entrance like what others had. Hello? Yeah. So yeah, so in 2018, like that chance was not like I mean that was lost, and then I went for medical entrance. So like everybody suggested, I studied for medical entrance, but my heart was still stuck up in genetics. Okay, so then my basics were very clear in biology, chemistry, and physics because obviously preparing for NEET exam is like we really have to begin from the scratch and then like make everything up to a mark. And then the year 2019 came, like I got into like MBBS college, and then I got an MBBS seat, but my heart was still aiming for something else because that was a private seat, and I really didn't want to spend that much money on a course which I was least interested in about. Obviously, saving others' life is a very good thing, but I really wanted a solid cure. Like more than healing, I really wanted to find how to find a cure for that healing. And then I applied for the same course in Calicut University and landed up there. And I was very fortunate that my basics were very clear because that 1 year gap which I took for entrance coaching really prepared me well and enough for choosing a career in genetics and be much more easy to tackle up things in a bit more easier way. So it was an earlier fascination with this DNA and inheritance, and then I chose genetics, like despite people questioning the scope and everything, because obviously we know that while thinking about genetics, because it was a new terminology at that time. I mean, a lot more people were not knowing. Like some were knowing, obviously, because like genes will be passing and everything. They know the surface level of thing, but they weren't sure that a country like India, do they really need genetics? They weren't aware about all this genetic counseling and all these like contagious disease and how this like inherited disorders are being passed on from one generation to another generation, and people were doubting that thing. But we should trust ourselves more than anybody else, isn't it? So I trusted myself, and I was firmly believing that the particular course which I chose will be something which will be having great impact in the coming future. And then my curiosity was something stronger than the conventional beliefs and everything. So I stood firmly with whatever I was taking ahead with. And my first exposure to scientific thinking in UG began in the subject research methodology. So I was fortunate with my HOD, Dr. P. Muralidharan, who was the teacher for research methodology. And he told us, like, right from the scratch, how we should curate a research, how we should state a hypothesis, and what is the difference between science and knowledge and everything. And that particular class helped me to like actually know about how a research has been done. And then like initially for a bachelor student, the PubMed, Nature, and all these sites are like a novel experience. So I started going through PubMed. I started reading research papers, which is really getting connected to what I'm interested in. So I had a like continuous familial inheritance of certain diseases like Parkinson's and cancer in my maternal family. So I was a bit more curious about why these conditions are happening. And there was also one guy in my class who was hemophilic, and I know that hemophilia is obviously a genetic disorder. So all these conditions are like something which doesn't have a proper cure till now, but researchers are under like all these studies and everything to find a proper cure even before the child is born, like just like mitochondrial replacement therapy and all, which we all might have heard, like all these like triple IVF techniques and everything. And then for my bachelor thesis, I did a work on Bacillus megaterium, which is basically a wastewater treatment bacteria, which is used for like with all the bioflocculation and removal of nitrogen, and with air, like an anaerobic bacteria, which can actually treat wastewater well. And that was my bachelor thesis. And it was, even though it was an independent work, we had a little bit of assistance from our guide, because initially we are bachelor students. So obviously we need somebody's strong guidance for this. And the UG experience that actually shaped me was strong theoretical foundation. That was one thing, because we had these subjects called medical genetics, cancer genetics, basic genetics, cytogenetics. Everything was interrelated with genetics. So even for developmental genetics, the entire developmental biology was studied in terms of genetics. And then in terms of cancer also, we studied every bit and then of genes, how genes are influencing the development of cancer. It was studied in an overview of genetics more than relating it with the basic biology. And then like learning the research basics, like obviously because genetics is a research oriented subject, so we really have to be focused upon the research basics. And then one of my first laboratory experience during my undergraduation was when I isolated DNA for the first time. Being a genetic student and being somebody who is really enthusiastic about DNA and how our entire information is being stored in this DNA, I was really fascinated to study about how do we isolate DNA. And the first DNA which I isolated was from a plant. I still remember it was a hibiscus plant using the CTAB like method. And when I saw the DNA right after adding the isopropyl alcohol and all, I was so happy, because in my entire family line, I was the only person who saw a DNA. That too, even though it's in a plant, it was a very novel experience and such a heartwarming moment. I still do remember the way I hold it with test tube with my hand, and then I was grateful for that particular moment. And then that obviously that initially developed every scientific mindset in myself, and then relating with model organism. So even in my UG class, we had this model organism culturing and all. So Drosophila melanogaster was cultured, and at that time only I realized that Drosophila melanogaster do have so many connection with human beings, because like in lot more cancer studies, neurodevelopmental disease studies and everything, we are using Drosophila melanogaster as a model organism. So for a common man, it might be a huge thing, because can you imagine like a fruit fly being a model organism for a human disease study? So if I tell this to somebody who is not from a scientific field, they'll be feeling it very odd. But us in the scientific community know that model organisms like C. elegans and this mice and Drosophila melanogaster really do have greater importance in like building up a new research hypothesis, isn't it? And then obviously being in a lab and being in a lab oriented subject, and much more a research oriented subject, we really do have many problems to tackle up. Like when we are doing a stain, simple mitochondrial stain, if the staining is not done properly, we may not be getting the exact result, but that mistake is something which will be tuning us into a greater success. So even if we fail for the first time, the chances of getting the second time getting 100% accurate is much more greater, and we know every aspect of that particular experiment. So when we fail in an experiment, that means that okay, next time we'll be finding it with some other new added source or something. So we'll be having a multi-dynamic source of like information about everything. Like we'll be thinking for every aspect in an experiment. So that was something which like taking genetics as my career helped me, because in a laboratory oriented subject, one thing we'll be familiarized with is that we'll be thinking from many other dimensions, like what if this happened, what if this happened, and how can I curate this, and how can I solve this. And all this really shaped me into something better in dealing with in human experience also. Uh, like for example, in life also we'll be dealing with different stuff, and at the same time we have this kind of mindset, like problem solving mindset, and we'll not be focusing more on the problem but more on curating the solutions. That was one thing which my undergraduation shaped me well. And then right after completion of my UG, I thought of moving on to either biomedical genetics or genomics. And I wrote the Central University entrance exam and then landed up in genomics, genomic science particularly in Central University of Kerala. So the major difference I found in genetics and genomics is that genetics is more like a single gene. Like I mean, it is focused upon the single gene, but in genomics, it will be like a large scale data involved. While studying genetics, we'll be studying about single gene involved in many such diseases and everything. But in genomics, it was like a large group of genomes coming together, like for a disease study and for human genome project. Everybody might have heard of the human genome project. So all these things like involve a large scale of data. Even in countries like Saudi Arabia and all, they are like now making things like a bit more like biometrics. They are now planning to do everything in a genome level, like uh, getting the data of a human's entire genome and everything. So all this is like the future, but we can clearly see that the future which we thought is actually being present, like things are happening in reality right now itself. So genomics and genetics are nearly not the future but the present itself. And the future of biology, like shifting into large scale data, is something which has been started in early years. Like this human genome project and all started long back, and it got completed after like so many trial and error methods and everything. And then all the consortium of people coming together and giving all the data and everything, finally that became a huge success. And now the precision medicine and genome sequencing is one thing, like even in hospital scenarios, even in crop development, even in precision medicine, even in any sort of thing where the genetics are involved, like in plant study, in drug development, everything, we can see that genome sequencing is one thing. Like for anything, we really do have a model organism, we do have to take a living material in which this study have to be done. So for that itself, we'll be doing all this genome sequencing and everything. So genomics is something which is a bit more advanced than many of other, like more than human genetics and biomedical genetics. Genomics is something which is looking onto a higher aspect, like in a bit more advanced manner. And then like being a top scorer of genomic science, I felt that it was not just because of theoretical study. There was like a lot more things involved, because in Central University of Kerala, internal marks was much more than external. Because internally we were assessed through not just theoretical exams but through internships, through seminars, through poster presentation, poster preparations, and then research rigor. That was the major things which they analyzed for internal marks and everything. So that was one thing. And more than shaping us as researchers, they will be shaping, I mean, shaping in such a way that we will be committed to our work. So commitment is one thing which every researcher should have. We should be really committed to what we are doing. In every job, in every industry, when we look at the most successful people, we can see that all of them are committed to what they are doing, and they have their 100% towards that particular work. And even if they fail in one thing, they will be finding it like another way to tackle it. And during my MSc journey, like one of the greatest fortunate thing I ever got was the mentors and the scientific environment at Central University of Kerala. And we had different labs, like mRNA labs, population genetics lab, metagenomics lab, metabolomics lab, plant genetics lab. And in everything, we had like ethical research committee and everything. And the research scholars were very cooperative, and even professors were like heartwarming, and they were like willing to do anything for students who have the passion for research. And I personally took immunogenetics and population genetics for my master thesis. It involved the MHC gene diversity of ducks. So I basically analyzed the genetic diversity of major histocompatibility genes in two different duck breeds, like one in Andhra Pradesh and one in Jharkhand. And I just curated the result like by analyzing their nucleotide haplotype diversity, how they are differing from one another, and if a disease like avian influenza or something affect them, which particular breed will be able to tackle it so much like at the earliest, and which one will be able to resist it. Because just for the for the for the duck breeders and everything, this study has been really helped them. It was an individual work, and then hands-on on molecular and immunology workflow was also being done, because the primer I used was for MHC particularly, and I was standardizing it for the PCR and everything. It was the entire thesis work was done by the usual process itself, like DNA isolation, then by quantification, then sequencing, then the interpretation of result through omega, cluster omega, and then AMOA, basic statistical analysis, and then their phylogenetic traits were analyzed and everything. And through them, I got a result, and then I published the data. And one more thing which I did did during my masters was summer internship in cancer cell culture. So moving from one field to another field was that I really wanted to get a basic exposure in everything, because everything is interrelated and interconnected. So getting a basic knowledge of cancer cell culture will be really helping in doing a work in immunogenetics, because I really wanted to move in a direction of immunogenetics, particularly focusing upon medical conditions. So cancer is one such disease which have a greater role in immunology as well, because when the immune gene defects get involved, it can really move into a field of cancer, isn't it? So I worked on melanoma cells and cancer cells and colon cancer cells, and I was assisting a research scholar who was working on a cytotoxic drug testing in a cancer cell. So the maintenance, and then how a media has to be prepared for the cancer cell, and then how it have to be like maintained, and then how to use the phase contrast microscope, because it was a novel thing. I had never used phase contrast microscope before. And then MTT assay, how to do an MTT assay, how to use multi-channel pipette, because the earlier I only used the single pipette, but using a multi-channel pipette was like using a very gigantic thing and then doing it with accuracy. So the precision and accuracy is one thing which we all should consider in research, because a minute error can sometimes cause a great deal of like cost and at the same time. And even for cancer cells, we really have to take care of it like babies, because the proliferation of the cells are huge. So trypsinizing was done. Trypsinizing is something which we do for cancer cells particularly, just to avoid the number of like increasing cells. And then this was the things which I done for my master thesis, and even I got exposure to advanced techniques and tools, which was like from Central University of Kerala itself, from different laboratories like RAPDSAs, ISSR, and genetic engineering laboratory was there, and that was a core study for us. So we had like 6 months training in genetic engineering as well, and then discovering immunogenetics. So the passion for immunogenetics actually built in like myself when I studied MHC genes in a bit more detailed manner. So while studying MHC genes, I was fascinated by the HLA genes particularly, because HLA is something which is interacting in us in human beings. So I was wondering which all loci does the HLA have, and I understood that they have this class 1 and class 2, then DP, DQ, DR, all these regions and everything. So I started reading papers for HLA, and I got really impressive of the work done by Dr. Moinak Banerjee, and he did a work in studying the cultural structure of Kerala population, that too by examining their HLA. And he was the first person, along with some other scientists, who did works in all this rape accused cases and victim cases and everything in Kerala, by analyzing their HLA particularly. So immunogenetics was something like we all know that the immune genes will be deciding how we fight against a disease, and at the same time they carry like millions of information in them. And then I did a training in Astamims that was under transplant immunology, and from there I got the idea of how the HLAs are involving in relating to the donor and the recipient. So in case of kidney transplantation and all, there like requires a lot more assessment prior to their kidney transplant, like actual procedure. So for that, they'll be doing HLA typing. Like they'll be using either oligonucleotides or using primers. That was the two methods. And apart from that, they'll be using NGS, like iron torrent, and using NGS method also, they'll be using the HLA typing. And That was a fantastic 1 month training I got from Astamims, and I really have to thank Dr. Vipin Vishwanathan for giving me that wonderful opportunity out there. And then I really got interested in how HLA and MHCs are related, because that have a very huge role in I'm in the related connection, because MHC is the parent of HLA. So basically this HLA have different regions, and all these different regions have multiple locus, and all these locus have different roles, and all these things have like different functions in our body. So basically looking onto an HLA, it will be like a very vast thing. So even when we're looking onto an immunology textbook, we'll be saying that there are like multiple markers and all these CD8 plus, CD4 plus, and everything. But earlier I thought that all these are just names, but now I clearly know that everything have a purpose in our body. And looking on to one thing and refining one thing will be taking so much time also in the same thing. So combining genetics and immunology is something which I would like to work for my near future, for my research study and for my doctoral study, and I'm preparing for that. And that's how I really dream to work in autoimmune disease and immune gene mapping. And so one thing which really made me choose research for my career aspect is unanswered questions. There are like a lot more unanswered questions, like why do that particular HLA is causing this particular disease, and or like why this DR region of HLA is having this particular conditions when a certain mutation act on our body, and how like the external environment, like for example the epigenetic modifications can really alter our body mechanisms. All these things are like unanswered questions, even though we have like answers for everything in all these AI models like ChatGPT, Gemini, and everything. There is not a like exact or precise answer for all these things. So for everybody, they will be stating a different hypothesis, and for everyone, they will be getting a different answer. So there is a saying which my professor told me during my master's thesis time. It was that when somebody is stating a science like a question in science, you cannot say that whether they are right or wrong, because a wrong answer can sometimes be right the next time, because every invention is getting novel and novel day by day, and everybody is inventing something new, and like more than inventing, they are discovering the things which were already been there. And another thing is like my desire to contribute to medicine. I really, like as I said before, I really love to contribute something for the cure, to heal something. And my passion for understanding all the mechanisms, and I love doing lab work. Like that gives me an immense pleasure. Find like during taking all this wet lab, dry lab, both computational and wet lab things. I'm really focused upon doing that, because that's a source of satisfaction which I get from doing the lab works, because for everybody they'll be having a passion towards something, and for me my passion is like doing all these laboratory works. Not just laboratory works, more than doing a technical thing. I really have like the result which we are waiting for is much more than this technical skills. So like, we'll be waiting for a final outcome. That final outcome is the final answer, and that joy after getting an entire good results, like just after like most of you might have done gel loading a gel in a PCR, and right after that we'll be looking through the gel electrophoresis, right? And if the PCR has been done properly, we'll be getting very good bands of DNA. That's the source of very relaxation as for a researcher. The same way when we're doing a laboratory work for like waiting for an end result, and the result came to be like something which we really looked up for, we'll be very happy. The same way uh, I'm like really passionate about doing that. And my long term goal is like getting something being done in immunogenetics, more than getting a PhD and the title of a doctor. I really love to do a research of my own, being an independent researcher under a well good laboratory, and with networking with a lot more people. And yeah, so during my studentship period, like for my bachelor's and my master's, during my final year of my bachelor's till my completion of master's, I was working as a online tutor as well, because I thought I really have to be independent myself in all terms. I really do not have to depend on my parents for the money which I spend for, or my books and my works and everything. Even for my mobile recharge, I didn't like care for asking them for money and all. So I started earning myself. I think we all should uh, earn yourself some money while attending the class at the same time, because we spend hours and hours time scrolling on our phone, and it only takes like 1 hour a day to earn very good amount of money till the like at the end of the month. So I started making a resume, like a very good resume, that included my communication skills and the way I mean, put about all these scientific terminologies and everything, and what I studied, and how passionate I am for teaching, like how passionate I am for making people understand about things. And then I made a resume and I uploaded in Internshala. Internshala was the first site where I uploaded this. And first company I worked was for Oriv Learning. So through Oriv, I got a very big exposure being an online tutor, and I worked there for 1.5 years. And after that, I moved on to Zeal Educators. And Zeal Educators was a company which is like dealing globally, and I was a tutor there in Zeal. And after Zeal, I worked in Connect My Expert, that's Telangana based company. In Connect My Expert, I was a full time tutor. So it was an online only, but I like freelance full time. So that means like whenever I had time, if the student is available, I can take class. So I earned my money through all these platforms. I think this can help you all in a great manner. Whoever is trying to earn money of their own, you can use these sources, and you can just prepare a very good resume and then start uploading it in sites. Now it's like Indeed, many more sites are there more than Internshala. Internshala was something exclusively made for students. But if you're a graduate, like undergraduate who have completed your studies, and if you're looking for a like part-time job or something, you can upload it in Indeed or Internshala. Even you can search for job positions in LinkedIn as well. And then like managed both my studies and my like this particular tutoring job simultaneously. I had plenty much of time apart from my like classes, so I spent time for that. And this job have really helped me in lot more aspect. Yeah, this job has given me like a lot more exposure to dealing with students, like getting to know about the current reality, like how this like school and everything environment are going on, and how the new changed curriculums are there. Because I was teaching different curriculums out there, like CBSE, ICSE, MYP, and certain international boards. And the NCRT textbook was the only thing which was the same as like old age. But in terms of ICSE and MYP and international boards, it was changing every year. So like students were getting a better exposure to all these things, and this will give us a bit more like knowledge regarding the new terminologies as well, because all their textbooks also have something which we studied during our basic elementary school time and all. So this will really help us to stay in a circle where we will be feeling grounded as well. At the same time, we'll be knowing the basics, and at the same time we'll be knowing the advanced techniques. So that was one thing which I did for my financial stability. And then I published a paper recently, a research paper that was a collaboration with Dr. Lavpreet Singh in Thapar Institute of Technology. I got this opportunity because of my networking through LinkedIn. So I attended a lot more webinars, and then reaching out to professors, obviously, because it's our need. So we really have to tell them, like our scientific rigor have to be mentioned them. So I started networking through LinkedIn, and then I started following professors. I started reading their papers, and then I started contacting them through emails. And then I asked scientific questions which I was having, and then through which I have connected with a lot more good professors across our country as well as in abroad. And then that paved a way for me for publishing opportunity. And my second paper, which is a first order paper, is it's been current under communication and about to get published. And the future of genetics and genomics, as most of you might be wondering, some will be having this like a skeptic thought of whether this is something which we really need, or is this something just a fancy course. But like we can say that when we are studying zoology, like or botany, I have heard from lot more friends, like lot of my friends, that they haven't seen a DNA in like in the real life. Like they are only doing research experiments like in terms of zoology, like a bit more animal specimens were be done, but it was something more like dissection and everything. And I don't know about the other colleges, but here out in Kerala, the DNA isolation was not being done for zoology undergraduates. And for masters, they were doing, but I'm not sure about the undergraduation. So in terms of genetics and genomics, one thing is that it's a research oriented subject. So obviously, things will be shown in a manner that how it has been in real. For example, DNA isolation has been done, RNA isolation has been done, plasmid isolation. All these things are being done in a particular way that we'll be getting to see everything what we are studying. So every now and then explanation, we'll be having this thought that okay, this thing can be done in the lab. So that kind of a thing, like that kind of a hope for seeing everything what we are studying has been real for me in this genetics and genomics. Like choosing this thing have been like a very good fortune for me. And one thing is that precision medicine, something like which our world has been looking on. I hope everybody might have heard about the CAR T therapy for cancer. It has been, it's like a 3 to 4.5 crore something cost um, therapy which is used for cancer healing. It's only be done in abroad hospitals like in John Hopkins and all. But few researchers in IIT Bombay in collaboration with ICMR and Tata have identified something like something sort of CAR T therapy, and they are being starting to introduce it to our country like India. So this has been possible only because of the advancement in all these careers. And we might be seeing that right after the COVID, like when COVID hit it, the genetics and genomics was the industry which has been blooming to a huge extent, because people went then focused upon like what is this variant, then what is this, like a bit more molecular biology aspect was being focused on. Like even a common man now know like which all variants of COVID has been passed. And all these things have been possible just because a contagious disease completely shut our country for like not globally affected for like 1.5 years. And then I think like the future of genetics lies in precision medicine, cancer genomics, obviously, like because day by day different types of cancers have been arising. And then AI plus genomics, because AI has been used for all these curations and everything, and this has been helping in advancing genomics to a huge extent. Then microbiome research is something which has been having a huge role, because the gut brain axis is something which has been studying for now. Like something like when we are eating something, and if the food is not having a proper ability, or if it is affecting the microbiome in a huge extent, that can really affect our brain, and that can really get down this neurotransmitters, and the dopamine, serotonin, oxytocin level can be reduced, and that can affect our mood. Now when we look onto the world, everybody, like one or the other person will be dealing with depressions, anxiety, stress and everything. All these are arising because of microbiome, and a proper study of microbiome research can really help in sorting out all these issues. And then synthetic biology, immunogenetics and all diagnosis, and these are the things which are having a huge role in the future of genetics and genomics. Like more than considering it as future, we can say that this is the like in present also we are under all these things. Like all these works are being done, like doing in different laboratories across the world, and all these are being possible only because of one single idea being developed in a researcher's mind. And that idea has been like, imagine somebody having an idea in their mind, and they curated an entire research hypothesis and proved it experimentally. And for example, a medicine for cancer, or a medicine for Parkinson's, a medicine for a very contagious disease, like even the vaccine, like Covaxin, Covishield, and everything, Pfizer, everything came to happen in reality only because somebody have a thought in their mind first. So that they made into a practical reality. So the same way, we all do have different ideas in our mind. It only takes one leap of courage to make it into a reality. So that's about future of genetics and genomics. And a career in this field will be PhD and research assistant roles. Like right after PhD, we'll be getting, either we can move on to academia, or we can move into industry, or the clinical genomics lab. Like there are like lot of plenty of labs in across our country who are like hiring for genome analyst positions and everything. Then for biotech and pharma research and development positions, bioinformatics, something which has been like very blooming, because every wet lab results have to be curated through bioinformatics data. And even a DNA isolation, one step, like I mean, DNA isolation earlier was like 2 day procedure, but right now it can be done with hardly 2 hours, because it is being made by chip based, and all this TrueNat and every other like methods have been making it a bit more easy. Like in hospitals and all, when we go, there will be like a single chip based mechanism, or a TrueNat machine for like isolating the DNA of all these HCV cases, all these meningitis cases, and everything. So they will be having different panels for the same, and through all this multiplex PCR and everything, they will be curating the result and giving out to the patients. Everything is getting on the higher level, and every now and then, all the results are being getting a bit more accurate, and everything is being evolving in a very huge rate. And scientific writing is and teaching is another career, like career opportunity here. And obviously, like new students will be willing to study, and obviously the teaching industry cannot be going down. And then facility core facility roles are also being there. So my advice for everybody who are currently doing their masters and their bachelors is that you all should follow your curiosity and not just pressure like from the society or from anybody else around you. You should follow what your mind says. And if your mind is telling you to stick on to the research, you really have to stick on to research, because this is not an industry which is going to be shut down for a night, overnight, or it has been falling for an overnight something. It has been something which is being like right from the mankind, like when man started all this education purpose and everything. From that time onwards research have been done, like even like some every now and then, you know, that all the like invention of the telephone or from the bulb or whatever it is, everything happened only because a researcher took a leap of hope in their work. And then build your skills like at as early as possible. Try doing all these courses in Coursera, edX courses, and then skill development, in sense that try attending all the virtual workshops, or even if you are able to attend all the conferences and everything, you can do that, because networking really helps you a lot. And then if you don't like do not have a LinkedIn account, I say that you all should start making a LinkedIn account as soon as possible, because we can connect with everybody who are there in your industry and your particular field, and you can get to know how their works have been done, and you will get new information and news about how everything is getting evolved across the globe. And you will be getting the opportunity to connect with every researchers around the world. And then be open to opportunities. Like if an opportunity come at your doorstep, do not just like drag it away. Just take that opportunity and start doing your work, because when you are like not believing in a like in your like particular thing, somebody else will be grabbing the opportunity. So better than that, just grab the opportunity which is coming in front of you. And don't be afraid of just putting off the unconventional paths. Like when people do have the tendency to follow what, like just like one ant following the other ant in the lane, we all do have this particular thing that following one person who have been successful in their life, but their map may be different, your map may be different. So just follow your heart, just think about how you can do or you can solve an issue. So just more than following up a conventional path, I would say that try doing something novel. Like even if you're taken a research oriented subject for your undergraduation or masters, just try doing something which can have a very good impact in for you as well as for the society. That's it. So any doubts or any questions from your side? Do anybody have to ask me something? Yeah, there are some charts. So Kriti Yadav, can you please tell me which supervisor you're talking about? I didn't understood what you're saying.
Participants, you can also unmute and interact with ma'am. We have opened the floor for Q&A now. You can ask your sessions from the session and also any guidance and tips that you would want her to share to you. So you can ask. This is your like chance now. You can even turn on your videos to interact and even you can turn on your microphones. You have the access. And those who have posted their questions in chat, you may just post them again to get those highlighted.
Yeah, so
Thanks.
like Pratima asked, like how to crack internships or research work in abroad university. So I would say you should make a very good CV by either using a Europass format or an ATS friendly format. ATS friendly is generally for every like even for both here and there. And try attending some online sessions. Like there are like plenty of YouTube channels which has been like explaining clear, up to date details of how to make a CV and all. Try making a very good CV, because your CV is the first impression. And apart from that, write a very good motivation letter, like why you want this. And then like search all the professors who are working in a particular field which you are interested in, take out their email ID from their either from the research paper or from their website, and then start emailing them, and you will definitely get a position. Okay, so other as you are a BTech biotechnology students, Kerala Agriculture is also study genetics for post graduation and research. Yeah, since you are a BTech graduate, obviously you can do that. You can write the Central University entrance exam and you can crack it. There are like I think for a BTech student, it would be a bit more easy for you to crack the entrance exam, and then you can join for genomics for your post graduation in Central University of Kerala. Yeah, Kriti. So regarding the immunology in terms of plant, the plant immunology is something very different from the animal immunology, and both are very diverse field. And in terms of plant immunology, we really have to study right from the scratch, like how the certain disease causing pathogens are affecting the plants, and how their cells are responding to these stimuli and everything. And something which I did is entirely different from plant. I'm not basically from a plant background, but obviously I can help you. You can reach out to me in my email ID. Uh, yeah. So genetic engineering, yeah. So for genetic engineering, it's something like incorporating a particular virus, or it particularly we'll be incorporating a vector to a particular genome for curating a new, like for vaccine developments and everything. And genetic engineering is something like for me, my professor was Dr. Padmesh Pilla. He's working in a metabolomics laboratory in Central University of Kerala. So the genetic engineering, we had this different essays, particularly the qPCR, RAPD assay, ISSR, and then like restriction cloning and everything. Then all these things were being under these particular subject, genetic engineering, and even we had this novel things like all this. Talent, all this fish, and everything comes under genetic engineering. It's basically a bit more advanced thing, because it is focusing upon a higher level of curating things in a bit more like transfection, transformation, all these things, like using just like CRISPR Cas9. I think most of you might have heard of CRISPR Cas9. So CRISPR is something we take this bacterial immunology, like the immune system, for like all this disease curing, and for several other things, like for using it as a media for vaccine development, and using it as a like an initial starter for everything, and for even for curing like all this color blindness. Even like lately I read a paper mentioning that for the color blindness, deuteroanomaly was being treated with CRISPR Cas9. So all these things are possible just because of later advancement in genetic engineering. So genetic engineering is more mainly focusing upon talent, CRISPR, and all these tools. That's it. And the PCR which I used for my like bachelor's sorry, for my master thesis was the conventional PCR thermocycler. But for the for the training which I done for transplant immunology, it was multiplex PCR. So it basically had like all like the reverse transcription, and everything was simultaneously going on, because since it's like hospital work, it has like the result have to be interpreted a bit more earlier than the research work, because like the results matter more than the research in terms of hospital and everything. But in terms of an academia results, like if we take 2-3 days more for interpreting the result, it doesn't matter as long as we have time. But in terms of hospital scenarios and the patient samples, we really have to make the results in a bit more faster way. Do anybody have any other question? Okay, so publishing a research paper, yeah, publishing a research paper is something that we really have to mark our potential in everything, right? For example, in scientific writing, we have to mark our potential. So research publishing a research paper means what? Our scientific writing has been accepted by an international journal. So that means the editorial board already know that, okay, this particular girl is having a bit more intelligence in writing things scientifically. So without, like, even now they have the tools for identifying all this AI and everything. So if we are like really focusing upon our work and writing it from the scratch with everything we have read through the review papers. By like even for review paper, we'll be like peer reviewed papers. We'll be analyzing a lot more papers and lot more works of the previous works. And when going through all these paper, we'll be having a bit more knowledge in that particular topic. So publishing a paper means we have a knowledge in scientific writing. So the skill for scientific writing will be increased when we publish a paper. So for pursuing a PhD for country like India, you really don't have to have a research publication, because here it's purely based upon fellowship and the interest of the guide. If you are like looking for universities like central universities, you can get into there with the university stipend. And if you are looking for a personal fellowship, you can write the JRF exam, CSIR NET, ICMR or GATE. Even if you are looking for a plant-based study, you can write ICAR exam, ICAR, and these exams will be giving you, and even DBT. So DBT exam will be giving you a fully funded fellowship until you complete your PhD, both for your JRF and your SRF. It will be covered. Yeah, of course. Like a medical, like completing a master's in medical genetics, you can obviously work in fertility centers. Just like you have to attend like 2 or 3 training in like embryology works and everything, because fertility centers more like they mainly work in embryology work, isn't it? So the precision should be very much accurate, because like all these methods are like doing with the high end machines and everything. So it's like very costly machines. So really, like really do have to have very much technical skills in that. Basically, there is not like any ongoing projects in for astrogenetics, I guess, in Central University of Kerala, because I'm a graduate of 2024. Until then, there weren't a project like that. Yeah, of course, it will be possible if you're pursuing a PhD and doing a part-time job simultaneously, because PhD won't be like a 24-hour job. It's mainly a job. I mean, in abroad countries, it's considered as a job, and here in our country also, it's a job. Like more than like a studentship, it's a job only. And here the thing is that for 1 year you'll be having a coursework, and rest of the years you'll be doing your individual work. And I think the best way to tackle up all these things are like start networking with scholars, like research scholars who are really working in PhD, or they are at the last years of their PhD. So they can really help you guys in all these things, because they'll be having idea about the work. And one thing is that we can do everything. Like even we can earn our money, we can start a life, and we can even study. But everything is that it's the way you manage your time, because we do have plenty of time, but like the priority which we give for our work is really do matter. So I think obviously you can do a PhD and start like looking for a part-time job as well. So yeah, as for my opinion, I would say that bioinformatics would be definitely a good opportunity rather than biotechnology, because bioinformatics is the future. More than biotechnology, bioinformatics really do matter. Start attending all this Python and R classes, get trained in that very well, and then if you're able to tackle up all this coding and everything, you can be a very good high-earning bioinformatician in the country, because bioinformaticians are paid more than biotechnologists.